Okay, this week's lab is another computer simulated lab. This time we're going to be using the nutrient pollution part of our Symbio software. So when you click on that, it's going to bring up a screen that looks like this. This is our lake ecosystem. And we're going to be looking at food chains throughout that ecosystem. We're going to have phytoplankton in our ecosystem. Phytoplankton are both green algae and cyanobacteria. They are both capable of undergoing photosynthesis, meaning they take in carbon dioxide and sunlight and are able to produce carbohydrates, and they give off oxygen. The phytoplankton can get eaten by our zooplankton, our Daphnia and our Bosmina. And the zooplankton can get eaten by our trout. So you first get to look at the different components of our ecosystem. Again, you have go buttons down here, you have a step button, and you have a step 52 button that will automatically proceed you through 1 year's worth, okay, of data in your stop button. So when we let our system go, the phytoplankton will be undergoing photosynthesis. The zooplankton are eating, the trout are eating, and some of our organisms are dying, turning black, and floating to the bottom. Those things that die and float to the bottom are going to have to get broken down or decomposed. Other things will decompose all of the dead algae and things at the bottom. All of your worms, okay? Fungi, all those ooey, gicky, yucky things at the bottom are going to decompose. When they decompose our dead organic matter, they consume oxygen in the process. So let's take a look back at our lake and see what's happened. We have these little binoculars that we can look at, and we can click on our different organisms. So our trout, the last prey that it ate was Bosmina. And we can then close that and we can look at, okay, our Daphnia. The last thing it ate was green algae. And we can click on the Bosmina and see the last thing it ate was algae. So that's the first thing that you're going to be looking at in exercise 1. Look at the organisms that are in your lake and see what they have eaten. You can find out the numbers of organisms in your lake by looking at the chart over here, or by left clicking on your mouse, and it will tell you the phytoplankton, there's 113 of them in your lake. It will tell you, okay, your cyanobacteria, there's 8 of those. And then your Daphnia, there's 10. Your Bosmina, there's 8. And then your trout, there's 5. And those numbers will fluctuate as organisms reproduce and die. In exercise 2, you're going to look at P, or phosphorus, in the water. And you're going to select that, and you're going to have a dissolved oxygen meter. That dissolved oxygen meter is going to tell you how much oxygen gas there is in that environment. Here's your phosphorus levels that you can change. Right now it's at 1x or normal, but through the course of this activity, you'll increase it to 2x and finally 4x. So when we let our simulation run, I'm going to click step 52, and it's going to let it run for 52 weeks or 1 year. All we have in our lake this time are our green algae and our cyanobacteria. And so it will automatically stop after 52 weeks. And you're going to record your green algae population and your dissolved oxygen. And so your dissolved oxygen is 7.1, your green algae is 177. And then you're going to increase your phosphorus input to 2x. And you're going to run your simulation again for another 52 weeks. Your green algae population is increasing, your dissolved oxygen level is decreasing. Green algae can undergo photosynthesis. They produce oxygen when they undergo photosynthesis. So when you are answering your questions, please do not say the green algae used up all the dissolved oxygen. What has happened over the course of the year is that green algae have a very short lifespan. They're constantly reproducing, dying, reproducing, dying. As their population numbers go up, more of them, are reproducing, but more of them are dying. They sink to the bottom and they have to be decomposed. The decomposers at the bottom of the lake are the organisms that are using up the dissolved oxygen in this experiment. You will continue your experiment by increasing your phosphorus level to 4x, and then you will do a graph on your green algae population and another graph on your dissolved oxygen level, and try to determine if there's a correlation between those two factors. It's going to ask you if phosphorus is a limiting factor for green algae. Keep in mind, a limiting factor will limit or control the size of a population. Therefore, if you add more of that factor or that nutrient into an environment and the population increases, that nutrient was a limiting factor for that population. So if you add factor A into that lake and population B goes up, factor A was a limiting factor on population B. When you added more of A, the population increased in size. You have other activities that you're going to work through. In exercise 3, you're going to look at dissolved oxygen. You're going to make some predictions about what's going to happen to your dissolved oxygen levels now that you've added trout into that lake. So remember, trout and zooplankton are going to eat some of that green algae. So that green algae population may not get as high. And if there isn't as much algae dying, maybe the dissolved oxygen level stays higher. So you're going to have to look at this whole ecosystem and what's happening. Trout will live longer than our phytoplankton. Eventually the trout will have to get decomposed, but they're going to live a lot longer than our green algae. Remember, it's the decomposers that use up the most dissolved oxygen in a lake environment. Then we're going to go on and we're going to look at what happens when we add toxins to our lake. We're going to add mercury to this lake. Mercury is a neurotoxin. Mercury will affect humans if we eat fish that have a lot of mercury in it. It will affect the development of a child's brain. It can affect adults' kidneys and organ functions. And if a woman is pregnant, that mercury can pass through the placenta to the baby and affect the development of the baby's neurological system. So we're going to look at what happens in a food chain when we add mercury. At time 0, Okay, we haven't added any mercury to this lake. So this little tool down here that kind of looks like a walkie-talkie, when you click on that, it will tell you each organism and the amount of mercury that's in it. So at time 0, none of our organisms have any mercury. Now we're going to let it run again for a year. Mercury is being added into this lake. And after our 52 weeks, when we click on, we now have mercury levels in all of our organisms. So our phytoplankton are going to take in mercury from the water. Those phytoplankton are going to get eaten by our zooplankton, our Daphnia and our Bosmina. They don't eat just one green algae, they eat a bunch. And so they start getting all of that mercury in their system. The trout are going to eat our Daphnia and our Bosmina. And they are going to start to build up mercury in their system. Biomagnification is when we start to see the highest levels of a toxin appear in the higher levels of our food chain. So when we do this again, another 52 years, we will see changes in the levels of mercury in those organisms. Now when we look at it, the highest levels of mercury are in our trout, because the trout is accumulating all of the mercury that the Daphnia and Bosmina accumulated. And they are accumulating mercury from the water and the green algae and cyanobacteria they absorbed. And so biomagnification, the highest levels of the toxins are in the highest levels of our food chain. And remember, people are going to eat the trout out of lakes. We can also get that mercury in us by eating the trout. You will then go through your number of years that you need to run. You'll record all of your population sizes and the mercury levels in your lab book. Remember, the population size you can find by your left button on your mouse and clicking over your organism. And you will then do a graph for each organism and the level of mercury that they have, starting at 0, then year 1, year 2, and year 3. And then the last activity you're going to do is called Mystery in the Lake. Your mystery in the lake is that you can change or alter the levels of both phosphorus and nitrogen. You are going to find throughout this activity that there was one species that was capable of nitrogen fixation. In other words, they can convert or make their own nitrogen. That species isn't going to care whether we add more nitrogen to that lake, because they can make their own. So you're going to look at the effects of phosphorus and nitrogen in those changes on our organisms. I'm going to suggest that you focus on the phytoplankton and their population sizes. In order to see the changes, you need to only change one of these factors at a time. So change first your phosphorus levels, and now you have options. You can increase them to 2x, you can increase them to 3x. Run your simulation and see what happens to the population of your green algae and your cyanobacteria. Then reset and change your phosphorus back, and now change your nitrogen. And again, you have options. Run your simulation and again see what happens to your two populations of phytoplankton. should be able to explain why when you increase nitrogen, one of those populations increases and the other decreases. And the opposite will be the case for the phosphorus, all based on their biology. You again will need to read through the directions in the introduction for this computer simulated lab. Again, you can complete the lab in stages. So long as you complete all of exercise 1, you can close out your software. Come back later and begin with exercise 2. Good luck, and if you have any problems, please email your instructor.